Hello and welcome to this series of lectures on understanding bias and introduction of variation as an evolutionary cause. A major theme of the synthesis era, going back to Fisher, was the rejection of theories alleged to be incompatible with Mendelian population genetics, including internalist theories that refer to internal tendencies of variation. For instance, when Simpson reconciled paleontology with this synthesis orthodoxy, he looked at apparent trends in the fossil record, argued that they probably weren't real trends, and concluded that even if the trends were real, they must be due to selection, because the only alternative is vital urges or cosmic goals without known mechanism. Where did Simpson get this confidence when generations of thinkers had simply assumed that internal tendencies of variation would cause evolutionary tendencies? We know the answer to this question. Simpson and the other architects of the modern synthesis believed that this kind of causal link was ruled out by the harsh mathematical logic of population genetics, because in fact, Haldane and Fisher had said that because mutation rates are small, mutation is a weak pressure, unable to overcome the opposing pressure of selection. It can only be important if mutation rates are abnormally high, or if selection is absent. Haldane argued this in 1927. Fisher followed up with a stronger version in 1930, and leading thinkers repeatedly cited this argument. So here are some examples that show the influence of Fisher. And this Haldane-Fisher argument continued to be influential in debates among evolutionary biologists. So for instance, in the seminal Developmental Constraints Review by Maynard Smith and colleagues in 1985, the authors actually raised the issue of whether developmental constraints cause evolutionary trends or patterns. And then they bring up the Haldane-Fisher argument as an impediment to this possibility. And they called for a reexamination, but actually they did not provide a reexamination. And so critics later complained that the advocates of developmental constraint had not provided any evolutionary mechanism. Even today, the concept of developmental constraint or bias is used without a causal grounding in population genetics. So for a final example, here is 75 years after Haldane's original argument. Gould explains, citing Fisher, that since orthogenesis can only operate when mutation pressure becomes high enough to act as an agent of evolutionary change, empirical data on low mutation rates sound the death knell of internalism. So by this argument, population genetics seems to place this do not enter sign on the door to internalist thinking. Because this Haldane-Fisher opposing pressures argument was so important, for instance, Provine identified it in this article about the contributions of mathematical population geneticists. He identified it as a key theoretical claim. Because this is so important, it's also important to understand that the argument is wrong. An evolutionary process that depends on events of mutation that introduce new alleles will be subject to biases in the introduction process by a kind of first-come, first-served logic that Haldane and Fisher did not address. This kind of causation, linking tendencies in the generation of variation with evolutionary tendencies by way of biases in the introduction process, was explained and demonstrated in 2001 using computer simulations of a simple population genetic model along with some mathematical arguments. And subsequent theoretical modeling has shown that this kind of causal link emerges under broad conditions. It's not a special case. Empirical studies have confirmed a key prediction, which is the effect of simple mutation biases like transition-transversion bias on the changes involved in adaptation. Contrary to Haldane's conclusion, this effect does not require high mutation rates or neutral evolution. And it's an effect of the introduction or origination process that Haldane and Fisher did not address. Instead, generations of theoreticians assumed that evolution begins with abundant variation and can be treated as a process of shifting gene frequencies, a process dominated by selection. Under this condition, all the alleles relevant to the outcome of evolution are present in the initial gene pool, and there are no biases in the introduction process, because there's no introduction process, thus there's no effective mutation bias. So the key to understanding this theory is to understand how mutation and selection can both be influential simultaneously in determining the course of evolution. So imagine a climber climbing on a rugged landscape. Not an ordinary human climber, because a human climber would scout a path to the peak. And we have to disallow that kind of foresight. So let's imagine a blind climber or a robotic climber that's operating by strictly local rules. And suppose the robotic climber moves by a two-step procedure. In the proposal or introduction step, the climber reaches out to grasp a nearby handhold or foothold. And each time this happens, there's some chance of the second step, that the climber will commit to this point of leverage and use it to move. So if we bias the second step, that commit step, such that higher points are more likely to be chosen, then this will cause the climber to ascend. The climber will go upward. But we could also imagine a lateral bias on the proposal step. We could suppose that the climbing robot has longer or more active limbs on the left side, and so tries more points of leverage on the left. And then the joint probability of proposal and acceptance will be greater on the left. So the trajectory of the climber will be biased not just upwards, but also to the left. If the landscape is rough, then the climber always gets stuck on a local peak, and it will tend to be up and to the left. If the landscape is a smooth hemisphere, the climber will just spiral upwards in the direction of the bias and end at the same place. If the landscape is constantly changing, like I've tried to indicate with this climbing treadmill over here, the climber will proceed until further change in the favored direction is limited in some way. The theoretical demonstration of the efficacy of biases in the introduction process now gives us a warrant to draw this kind of green arrow here connecting cause and effect, and to propose mutational hypotheses for patterns on this basis. Recent empirical work that I'll summarize in a moment establishes that biases in the introduction process are a true cause of orientation or direction in evolution. So for the remainder of the talk, I'm going to focus on what is new and interesting about this line of argument. So first, we have a body of empirical results, and these stand by themselves in the sense that the effect of mutation biases can be evaluated prior to any theoretical considerations. The key to understanding why these results matter, however, is to consider the mutation pressure theory from Haldane and Fisher, in which the influence of mutation depends on high mutation rates or neutral evolution. By contrast, the efficacy of biases in the introduction process does not require neutral evolution or high mutation rates. Instead, under this theory, merely quantitative biases in ordinary types of mutations, so like transition-transversion bias or GC bias, can influence adaptation. That's a distinctive prediction of this theory. So the empirical case is based on results of laboratory adaptation or cases of natural adaptation in which an adaptive phenotype or behavior has been traced to molecular changes with verified functional effects. And some of the papers are listed here. Some of the data come from famous cases of adaptation traced to the molecular level. So for instance, spectral tuning, cardiac glycoside resistance, hemoglobin adaptation in high-altitude birds, tetrodotoxin resistance. This is about parallelisms in ruminant ribonucleases, and this one is about parallelisms in echolocating bats and whales. So if we gather the data from all these different excellent analyses of adaptation traced to the molecular level, what we find is that there's a bias toward transitions. So transitions are these mutations that happen usually several-fold higher than transversions, and there's twice as many transversions, so the null expectation is 1 to 2. But when we look at cases of parallel adaptation in the laboratory, we see a four-fold or maybe 6 or 7-fold, depending on how we count them, excess of transitions. And when we look at natural cases and aggregate all these data so that the numbers are large enough to do statistical tests, we find about a 2-fold or a 3-fold bias favoring transitions relative to the null 1 to 2 ratio. So that's the empirical case. It may not sound very glamorous. I'll explain later why I think it's important. So secondly, we have a useful theory to apply to understanding evolutionary patterns that might have internal variational causes. So here's a kind of general framework for thinking about that. On the left here, we have the mutational and developmental causes of tendencies of variation. And in the background, while those causes are operating, there are environmental conditions which can affect mutation and development. And then there are broad features of the architecture of state spaces that sometimes come into play as formal causes of certain types of biases. And on the right, we have the evolutionary explananda, the things that we want to account for. So some favorites of mine, like patterns of parallelism, directional trends, self-organization in the sense of findability. I'll talk about each one of these things along the way. So what are some other theories that specify a role for variation that we might want to compare? So there's Darwin's, I think these are the 5 most important listed here in chronological order. So Darwin's theory that the laws of variation bear no relation to the structures built by selection says that there's no dispositional effect of variation. It's just raw material, it's just substance. And selection provides all the shaping and all the creativity. There's a very old folk theory that says that variation imposes hard limits on selection, or it sets out a list of possibilities from which selection chooses what it wants. There's the mutation pressure theory of Haldane-Fisher that says that mutation acts by driving alleles to high frequencies. There's the theory of evolutionary quantitative genetics, and then there's this new theory of biases in the introduction process. So for instance, if we put Darwin's theory in this context, Darwin believed that there were laws of variation, that variations emerged by what he called immutable laws, but they bear no relation to the structures built by selection. So this theory puts a brick wall there. Variation is not a dispositional cause under this theory. Instead, selection is the only dispositional cause. Excuse me. The mutation pressure theory kind of makes a hole in this wall, but it's not a very big hole. The theory can explain things, patterns in molecular evolution, for instance, but only if they involve neutrality or high mutation rates. The folk theory of hard limits is invoked a lot in the literature, but I don't think it's a very useful theory unless one interprets it loosely. It can't explain those effects of transition-transversion bias that we talked about previously, because they're not hard limits. It's just a quantitative bias. It doesn't explain the findability phenomenon that we'll discuss a bit later. Evolutionary quantitative genetics does provide a limited grounding for internalist thinking, and it's sometimes invoked in the evo-devo and evolvability literatures, with important work from authors like Thomas Hansen or Günter Wagner. However, it doesn't provide a way to rebut the Haldane-Fisher argument or explain molecular parallelisms or offer an account of directional trends or molecular findability. So let's use this framework to consider a comparison. So here on the right, we have some observed patterns of molecular evolution. Maybe we're talking about patterns of parallel change, or we're talking about some other kind of pattern. And we can ask, what's the effect of GC bias or transition bias or something like that? Most of the theories just don't apply. So neo-Darwinism is irrelevant. These are not hard limits, so the hard limits theory doesn't apply. Evolutionary quantitative genetics doesn't apply. So we're really just down to two theories here. There's the pressure theory, and there's biases in the introduction process. The literature of molecular evolution refers to the pressure theory and assumes that when mutational effects happen in evolution, that neutrality is required or implied somehow. So here's an example of that from Lefay. They're talking about changes in amino acid and codon compositions, and they say they represent neutral sequence change that has been caused by strong strand and species-specific mutation pressures. So if we're specifically considering adaptive changes like adaptive parallelisms, then the theory of biases in the introduction process is the only theory that we have if we're looking for mutational explanations. What about developmental constraints? As mentioned previously, the developmental constraints literature typically presents a gap in causal reasoning, and that goes back to that original Maynard-Smith paper that we talked about. But it also continues in the contemporary literature. So if we wanted to assign a causal interpretation, we could invoke hard limits, but that theory is not very useful. Or in some cases, we can invoke evolutionary quantitative genetics, and some constraints claims refer to that framework. To understand how the efficacy of biases in the introduction process suggests a way to rethink this issue, we first need to understand how genotype-phenotype maps induce biases in the mutational accessibility of alternative phenotypes. So the genetic code is the genotype-phenotype map that relates codon genotypes to amino acid phenotypes. Most organisms have 1 million or 10 million characters that develop according to this genotype-phenotype map, which doesn't change, which makes it a good case to study. So starting with the methionine phenotype, there are 4 different amino acid phenotypes that are accessible with a single nucleotide mutation. Leucine is accessible with 2 different single nucleotide mutations. Isoleucine is accessible with 3 different single nucleotide mutations, and these amino acids shown in gray, like cysteine, can't be accessed by any single nucleotide mutations. They require a double or a triple. So in this way, the genotype-phenotype map induces 4 different degrees of mutational accessibility of alternative phenotypes, starting from the focal phenotype. Even if each rate of mutation from one codon genotype to another is the same, there will be 4 different rates of introduction of alternative phenotypes. So this is the local effect of accessibility, the effect of mutation from. But there's a second global effect of mutation to, by considering all the arrows that point to something rather than what points away from it. So for instance, reverse all those arrows and you get all the mutations to methionine. if we look at all the mutations to leucine, there's going to be more because leucine has more codons. And in general, the phenotypes that have more genotypes in genotype space have more mutational arrows pointed at them, which makes them more findable by an evolutionary process that searches possibility spaces by mutation. And this expected effect is actually seen in evolution. It's well known in molecular evolution that there's this correlation, and this goes back to the 1969 article by King and Jukes, that the frequency of amino acids in protein sequences correlates strongly with the frequency expected from applying the genetic code to randomly generated sequences. Most of this effect comes from the numbers of codons in the genetic code. So methionine and tryptophan down here have 1 each. Serine, leucine, and arginine, which is a bit of an outlier, have 6. Isoleucine has 3. These guys up here have 4, and these guys down here have 2. More generally, any non-trivial genotype-phenotype map is going to induce these same 2 kinds of effects. So this is a stylized genotype-phenotype map showing how 3 different RNA folds are distributed in sequence space. And the global findability effect is a function of the total contour length or surface associated with a phenotype network. So in this example, the gray phenotype P0 is twice as findable as the other 2. It has twice the contour length. That's the global effect. But P1 and P2 have the same findability, but starting from P0, P2 is much more accessible. That's the effect of local accessibility. So starting from P0, P2 is much more findable or much more locally accessible because it has more shared contour length. Okay, to summarize then, we can consider the theory of biases in the introduction process as a basis to construct causal models for effects of developmental biases in variation, and this seems particularly useful for discrete traits. Likewise, the theory provides a basis for effects relating to the abundance and connectedness of phenotypically defined networks in genotype space. So these effects are typically described in the molecular evolvability literature in terms of background conditions or in terms of self-organization. That was Kaufman's term, self-organization, rather than being ascribed to a population genetic cause. A few years ago, R. Lewis's lab did population genetic simulations to show that these effects arise from kinetic bias due to mutational introduction. They are not an effect of selection, which is what Kaufman argued. So the arrival of the frequent for Schopper and Lewis means that folds that occupy a greater volume of genotype space have more mutational arrows pointed at them, which makes them more findable by an evolutionary process of exploration based on mutation. Okay, biases in the introduction of variation can influence the course of evolution, or biases in variation, I should say, can influence the course of evolution by inducing biases in the introduction process. Why was this influence not recognized sooner? Why was it absent almost completely from 20th century evolutionary discourse? Why was it absent from these key topics listed here, all of which we've touched on directly or indirectly? This kind of why question can have many different answers. The kind of answer that emerges from studying mathematical models and formal arguments about causation is that the action of biases in the introduction process falls outside of a very influential paradigm, the shifting gene frequencies paradigm, which says roughly that evolution can be understood in terms of shifting the frequencies of alleles present in an initial population in an initial gene pool without new mutations. The experimental touchstone for this view is Caussal's experiment with hooded rats. Causal was able to breed nearly all black and nearly all white rats in just 20 generations of selection, which was not enough time for new mutations to have played a role. Johansson's earlier selection experiments showed only that selection changes the frequencies of pre-existing types, but Caussal's experiment was interpreted to mean that selection can create new types just by shifting the frequencies of genes in an initial gene pool. That's why Caussal's experiment was so important for the modern synthesis. That's why it plays such a central role in Provine's historical analysis. That is, this result, the Causal experiment, is important logically and it was important historically because it allows for a rebuttal to the mutationist conception of evolution as a two-step process in which mutation proposes, selection disposes. Now, if all the alleles relevant to the outcome of evolution are already present in the initial population, then mutation does not introduce new things, but just shifts the frequencies of existing things. But mutation rates are small, so they can be ignored if you're working within this paradigm. That is, theoreticians within this paradigm could explore the dynamics of evolution without worrying about mutation rates. For instance, I never tire of citing this short treatise on mathematical population genetics by Edwards from 1977. There's hundreds of equations in this book. None of them has a mutation rate. The term mutation only appears on page 3, where the author tells us all genes will be assumed stable and mutation will not be taken into account. Now, this sounds crazy, but I'm not the only person who's noticed this. I'm going to quote some authorities for you here. So Yedid and Bell say that there's a short-term process where selection merely sorts the variation already present in the population, and this process is described by the mathematical theory of population genetics, which begins by defining a fixed set of genotypes and cannot provide a satisfactory analysis of the second, longer-term process where quite different genotypes arise. Hartland Taubes say that there's two kinds of models. There's the classic kind of model where the alleles are stipulated to be present in the initial population. And then there's the kind of new molecular evolution models which emerged in 1969 where new mutations happen, but they're neutral or nearly neutral. And here Eshel and Feldman are drawing a distinction between short-term evolution that assumes a fixed set of genotypes and shifting gene frequencies, and then long-term evolution where new mutations are tried out. And they conclude that since the time of Fisher, an implicit working assumption in the quantitative study of evolutionary dynamics is that qualitative laws governing long-term evolution can be extrapolated from results obtained for the short-term process. Now, each of these sources that I quoted here is suggesting the same two things. The shifting gene frequencies paradigm of evolution without new mutations was a limiting paradigm in 20th century modeling, and it was breaking down at the end of the 20th century because these authors were bringing attention to that. Now, this rethinking of evolution as shifting gene frequencies was not just a mathematician's trick. It wasn't just a convenient guide for modelers. It was part of a wide-ranging paradigm. So Richard Michaud, in this 1981 paper, identifies the shifting gene frequencies as a paradigm in the style of Lakatos. He calls this the hardcore of population genetics, is that the species is a point in gene frequency space, and evolution is movement in that space. This point of view, he says, is the basis of the population genetics approach to evolution. What does that mean? Well, historically, prior to the synthesis era, there were no agreed answers to big questions like, what is evolution? What are the causes of evolution? How do I study them? And so on. And the shifting gene frequencies paradigm provides crystal clear, wide-ranging guidance on how to think about and how to study evolution. Here's what it says. What is evolution? Evolution is shifting gene frequencies. It's not just an event of mutation or an event of birth or death. It's an aggregate population level shift. Where does evolution take place? What's the locale of causation? Evolution happens at the population level because populations are the things that have frequencies. How do I model evolution? What's the state space? Well, the state space is an allele frequency space, like Wright said in that statement. What are the causes of evolution? Because evolution is shifting gene frequencies, the causes of evolution are the processes that cause shifts. Selection, drift, migration, mutation. A process that causes larger shifts is a larger force. Selection is the strongest force. Mutation is the weakest force. How do I study causes? I'm sorry, paleontologists. I'm sorry, developmental biologists. But the only way to study causes is by observing populations as they undergo shifts in frequency. That's the answer provided by the shifting gene frequencies paradigm. Now, some of these answers became explicit doctrines, others became hidden assumptions and soft prejudices, and they exerted a powerful influence on formal reasoning, although it's always the case in evolution that there's all these fuzzy verbal arguments and people are all over the place. They're not constrained by these things. So, for instance, Meyer clearly would not have agreed to saying that evolution is shifting gene frequencies. He didn't want to define evolution in that way by reference to the genetic level. So with this shifting gene frequencies theory as context, let's look at the forces theory in more detail. So at the population level, we can ignore individuals and represent frequencies as continuous valued variables. So here's one dimension representing the frequency of A2 as opposed to A1. The population is a point on this line. It has a position, and movement of this position is evolution. The processes that cause shifts are the evolutionary forces, and the currency of causation is a small shift. So here, going from frequency F to frequency F plus delta, a small shift here. That is the increment of evolutionary change. So any force could cause this change. Selection could cause this, drift could cause this. This is a pretty big change for mutation because mutation rates are small. Mutation is indeed a weak force, but in principle it could cause each of these shifts. So consider the opposing pressures argument. We can apply this, for instance, to a monogenic disease-causing allele, which is what Haldane did. So the force of mutation is pushing the frequency upward. The force of selection is pushing it downward. They reach a balance, and that mutation-selection balance frequency is much closer to 0 than to 1 because mutation rates are so small. So if we extend this to 2 dimensions, we've got a 2-dimensional space. Evolution is a path in this space. We can break that path down into increments, and each increment could be caused by any force or by any combination of forces. That's how the forces theory achieves generality. It allows us to reason consistently about any point in this space, except for one enormous problem. It doesn't work at the edges. The logic of the forces theory only holds together for systems that remain in the topological interior of an allele frequency space. So here's a shift that's the same mathematically in these 2 figures here, but all I've done here is I've just moved it down to the bottom axis, and now it's a shift upward from 0. Selection can't do that. Drift can't do that. In this trajectory, you have to have a mutation event somewhere that introduces an allele changing the frequency from 0 to something greater than 0. What this means is that in the interior of an allele frequency space, mutation is a weak force that typically can just be ignored. But at the surfaces of an allele frequency space, mutation has a power that is infinitely greater than selection and drift. It has the power to introduce something new. That is, a qualitatively different kind of cause must operate at the surface of an allele frequency space. And the name that we give to this qualitatively different kind of cause is the introduction process or the origination process. So for 150 years, critics of Darwinism have complained that selection can't create anything new, and proponents have responded either by saying, well, selection creates new and improbable combinations of things that already exist, or they've said that creativity can be a matter of just shifting existing quantities to make something new. And those arguments may have some value, they may have some applicability, but they just break down when we're talking about evolutionary genetics, because in fact, we can't get the dynamics of evolution right without an introduction process, a point process that introduces new things. We know that now. It's been shown. That's the most important thing I can say to you today. And before leaving this topic, I just want to stress again, there's this big cloud of fuzzy verbal arguments. So when I talk about the shifting gene frequencies paradigm, I said it emerges from analyzing mathematical models and formal reasoning. If we look at informal reasoning, it's just a mess. There's a lot of gibberish. Another novel implication to discuss is that we can specify a greatly expanded causal grounding for internalism. So what does that mean? Well, let's begin with the lack of a causal grounding. So for instance, the argument that development cannot be construed as an evolutionary cause was deployed very effectively against evo-devo by Ernst Mayr and others in the 80s and 90s, going up to Mike Lynch more recently. And this argument follows from the shifting gene frequencies paradigm because development is not a mass action pressure on allele frequencies. But now we can rebut this argument. We can say, no, Ernst Mayr, we can construe development as a cause because the developmental genetic organization induces phenotypic biases in exactly the way that I described, and those can act as biases in the introduction process, which is a known mechanism. And more generally, the lack of a causal grounding manifests in a variety of ways. So we've already discussed this classic debate about potential causes of direction. In regard to evo-devo, we have both the objections of critics and the explicit search for causal legitimacy by evo-devo proponents, so reflected in arguments about causal completeness and lineage explanation. And similarly, in regard to intrinsic sources of form or order, we have both the struggle of structuralist proponents of these ideas to articulate a causal basis for their views, and the objections of critics, such as one reviewer of Kaufman's 1993 book, who said that his references to self-organization were almost magical. So an adequate causal grounding would require not just an abstract theory, but a way to rebut objections and a way to demonstrate efficacy, so we can get away from this objection of lack of evidence. And today we can supply all of that. So the influence of mutational and developmental biases and variation by way of biases in the introduction process provides a way to understand the key themes of internalist thinking. So taxon-specific propensities. We can look at the classic study by Allberg and Gale, where they proposed that salamanders and frogs have different taxon-specific propensities for losing digits, and we can propose a causal basis for that in terms of developmental biases in the introduction of variation. We can specify a causal basis for this findability argument about intrinsically likely forms, and although I haven't gone into this, we can do the same thing for rationalizing the idea of directional trends. In addition, we can rebut objections. We can rebut the Haldane-Fisher argument. We can rebut the causal status objections because these all rely on the shifting gene frequencies paradigm, and they fall apart when we recognize the efficacy of biases in the introduction process. We can provide a response to the causal completeness argument that goes through population genetics rather than trying to go around population genetics with lineage explanation. The aspects of development that you would use in lineage explanation to rationalize the developmental genetic plausibility of an inferred transition are the same aspects that you would apply to hypothesizing developmental biases in the introduction process within a context of population genetics. And finally, we have this empirical demonstration of efficacy and scope. Now, I mentioned previously, this argument does not seem very glamorous, this effect of transition bias. It's definitely not the most fascinating aspect of adaptation. However, we can use it to show something that wasn't shown in 40 years of evo-devo. So this is like that classic argument from Max Delbruck about a bacteriophage or the hydrogen atom of genetics. So the hydrogen atom with one proton and one electron, that's the simplest complete physical system that you can use to interrogate the basic laws of matter. And at the dawn of the molecular biology revolution, Delbruck chose phage as the hydrogen atom of genetics, the simplest complete system that you could use to interrogate basic laws of genetics. So my colleague David McCandlish recently drew on this trope to say that these simple cases of mutational biases acting on the amino acid changes involved in adaptation, those are the hydrogen atom for internalism, the simplest systems that we can use to interrogate the influence of biases and variation on the natural process of adaptation. Okay, and finally, wrapping up, we have the basis for a fresh perspective on history and on current disputes in evolutionary biology. So, as I mentioned, a formal theory for the efficacy of biases in the introduction process was absent, so far as anybody knows from the 20th century discourse, absent from key issues like this. And so what we have here is not a story of modern synthesis triumph, but a story of failure. And this failure is linked very strongly to the shifting gene frequencies paradigm, which I would argue is important for understanding other challenges as well. So I think sequence comparisons, but not the shifting gene frequencies theory, induced immediately a conception of evolution as a Markov chain of origin fixation events. And when I say I mean the biochemists who made these sequence comparisons immediately began to take on this kind of mutationist view of evolution. And then the neutral theory emerged as this overly narrow solution that essentially tied mutationism with neutralism. The alleged resolution for the macroevolution debate of the 70s and 80s was to take that population level and add another level of populations of species above the level of populations of individuals. I've already talked about evo-devo and self-organization. Most of the extended synthesis themes have a foundation that goes outside of the shifting gene frequencies paradigm. So I suppose one could draw a number of different lessons from this, but the lesson that I draw as a scientist is to ignore the traditionalists who've been defending tradition by making everything woolly and flexible, and to focus instead on theories and formal reasoning. As again, there's a big cloud of noise and argument in evolutionary biology, but most of this is informal and it's politicized, and analyzing it, I think, does not hold much value. But we can find deep structures in evolutionary thinking that come from theories and paradigms, and these deep structures they emerge most clearly in formal arguments, especially, I think, when they're used to reject possibilities. All right, to summarize, I've described a new and I think widely applicable theory for a dispositional role of mutagenesis or varigenesis. Because we can make an empirical argument for its importance, it allows us to argue that this is a known mechanism and we can use it to specify a causal grounding for internalism that did not exist previously, that's quite broad, and that allows us to rebut classic objections. it represents this theory, an exception to the forces theory and to the shifting gene frequencies paradigm. And these exceptions and this new causal grounding, together all of this provides a new perspective on historic and contemporary debates. So thank you very much for listening. Please watch this channel for more talks on this theme as they emerge. And I'm now going to silently scroll through the bibliography for those who are interested in sources. Again, thank you.